What is that fundamental spark that separates something that's alive from something that's not? It is one of the biggest questions in all of science. And today, we're going to dive into a really fascinating way of trying to answer it. Not by taking life apart, but by actually trying to build it from the ground up on a single piece of glass.
This is the big one, right? At what exact point does a random collection of molecules stop being just chemistry and start becoming biology? It's a question that really blurs the line between science and philosophy, and it's the mystery at the heart of the research we're exploring today.
Okay, so to even begin to build life, you've got to have a blueprint, right? A working definition. So before we even get to the lab bench, we have to ask a more basic question. What are the essential ingredients that scientists can all agree are fundamental to any living system?
Well, when you boil it all down, the classical definition of life usually has 4 key properties. It has to be able to replicate itself, it needs some kind of metabolism to use energy, it has to be able to evolve, and it needs a container, a boundary that separates its inside from the outside world.
Now here's the key insight, and it's a really important one. 3 of those properties, replication, metabolism, evolution, they're all processes. They happen over time. But the compartment, that boundary, that's a physical thing. It's a place. And that makes it the most logical starting point if you're trying to build life from scratch.
So that container, that essential boundary, is what we call the cell membrane. It's basically the wall that creates an inside that's totally different from the outside, and that's what allows all the incredibly complex chemistry of life to actually happen. But how did this crucial container first get connected to life's information? It's code.
And this leads us to a huge debate in biology. On one side, you've got the RNA world idea, which says information, like self-replicating RNA, had to come first. On the other side, you have the lipid world idea, which argues that the container, these simple little bubbles of fat or lipids, had to form first. So what was it? The code or the container? This research is all about trying to connect those 2 worlds.
So how in the world do you tackle a problem as massive as the origin of life? Well, you simplify it. You take it out of a messy, complicated, 3-dimensional world and you put it onto a flat, controllable surface where you can actually watch every little interaction.
Yeah, you could think of this whole approach like a test kitchen, but for the ingredients of life. What these researchers did was spread a super thin layer of lipids, that's the stuff cell membranes are made of, onto a glass slide. This creates a simple 2-dimensional version of a cell's surface, which is the perfect little sandbox to add other ingredients and just see what happens.
All right, let's get into the nitty-gritty. We're going to walk through the 3 key experiments they ran on this 2D membrane. Each one is a step closer to understanding how life's machinery actually works, and they move from using all-natural parts to completely synthetic ones.
First up, reconstruction. The goal here was pretty straightforward, but really clever. They wanted to see if you could get lifelike behavior just by taking life's own natural parts and putting them back together. So they took lipids that they extracted directly from an amoeba's cell membrane and mixed them with the amoeba's own cellular guts, which is called the cytosol.
So here's what that setup looks like. You've got the lipids from the amoeba, spread out nice and flat on a surface. Then the researchers inject the amoeba cytosol, all of its internal proteins and machinery, right on top of this lipid layer. The big question is, can these components, even when they're separated and then recombined, still produce lifelike behavior?
And the answer was a huge yes. It was incredible. On the left, you see the flat, kind of lifeless patch of lipids. But after adding the cytosol, look at the right. The membrane just buckles all on its own, rising up from the surface and forming this complex 3-dimensional shape. This is a key part of how cells move, and it happened just by adding the cell's internal machinery.
Okay, so we know the natural parts work together. But is there something kinda special or even magical about those specific lipids from the amoeba? The next experiment, constitution, tests exactly that. They swapped out the amoeba's custom lipids for some generic, off-the-shelf ones to see if the membrane's recipe actually matters.
So the setup is almost identical, but with one crucial change. Instead of that complex, messy mix of lipids from a real amoeba, they use a perfectly clean, uniform membrane made of just a single type of purified lipid. Then they add the exact same amoeba guts from before. The only thing that's different here is the container.
And the result was completely different, and honestly, pretty shocking. Instead of buckling into a cool, dynamic shape, the generic membrane gets totally torn apart. Those same cytosol proteins that built structure before now just punch these huge holes in the membrane. So the crucial point is this: the specific composition of the membrane is absolutely essential. It's not just any container, it has to be the right container.
And this all leads to the final, and I think the most ambitious experiment, construction. If the first experiment showed that natural parts work together, and the second showed how important specific parts are, this one tries to build a brand new system, totally from scratch, that directly connects that lipid world container to the RNA world code.
And this is just, it's so brilliant. The researchers designed and then synthesized a hybrid molecule. Just look at this thing. One end is a lipid, which is designed to stick itself in the membrane. That's the container part. The other end is a short strand of DNA. That's the code. They literally physically linked information and structure into one single thing.
And here is the payoff. By applying a super simple external trigger, just a little bit of heat, they could make the DNA part of the molecule change its shape. And because that DNA is physically tethered to the membrane, this change in information forces a change in the physical structure. The flat 2D membrane spontaneously pops up into a 3D structure. They directly controlled structure using information.
Okay, so let's zoom back out. What does all of this actually mean? We've seen membranes buckling, getting holes punched in them, and even information-driven shape changes all happening on a tiny piece of glass. Let's connect these amazing results back to that huge question we started with.
You know, this diagram from the research really captures the whole grand vision. Down at the bottom, you have the simple elements, the lipids, the DNA, the proteins. And way up at the top, you have these big abstract states of life and non-life. The whole goal of this bottom-up approach is to map out the rules and the pathways, all those arrows, that connect the simple micro-interactions to the macro state that we recognize as being alive.
And really, that's the big takeaway here. These experiments aren't just about recreating lifelike behaviors in a lab. They're about learning the fundamental design principles of life itself. By becoming molecular engineers, these scientists are methodically and literally blurring that line between a puddle of chemicals and a living system.
And that leaves us with a final fascinating thought to chew on. If all of these dynamic, lifelike behaviors that we see can just emerge from the interaction of a few carefully chosen molecules that are just following the laws of physics and chemistry, what does that say about the nature of life itself? As we get better and better at building the machine, we get that much closer to understanding the ghost within it.